Consumers are rediscovering the good taste of smoked fish. Smoked fish can be profitable, whether it's sold as an ingredient or as is. You may decide to start producing smoked fish to meet this growing demand, but don't, until you learn how to do it safely. Smoked fish is a potentially hazardous food. Mistakes made during fish smoking or improper storage of smoked fish can be deadly. However, if you do certain steps correctly, your smoked fish will be delicious and safe. All fish processors, including those who smoke fish, are required to implement the Hazard Analysis Critical Control Point, or HACCP system. This system is designed to ensure that fish products are safe. Under HACCP, certain steps called Critical Control Points, or CCPs, are monitored and recorded to control identified food safety hazards. This video will teach you about the food safety hazards associated with smoked fish. You will also learn about the steps considered to be CCPs for fish smoking. Many states have specific regulations about making and labeling smoked fish. To minimize confusion, this video will cite guidelines available from the United States Food and Drug Administration whenever possible. In some cases, the video will describe state regulations from Wisconsin and Michigan. Before you start smoking fish, become familiar with the appropriate regulations in your state. To obtain copies of these regulations, contact your state government or extension specialists at your state's land-grant university. Hazards associated with any fish or fishery product may be biological, chemical, or physical in nature. Biological hazards include disease-causing bacteria, viruses, and parasites. Chemical hazards may be environmental contaminants such as pesticides or industrial chemicals. They also include aquaculture medications that aren't used correctly. Pieces of metal, wood, or other foreign objects are physical hazards. By far the most significant hazard associated with smoked fish is a bacterium called Clostridium botulinum. This bacterium produces the most poisonous natural toxin known, the botulism toxin. If not treated in time, the botulism toxin can cause death by paralyzing the muscles used to breathe. Clostridium botulinum is found in underwater sediments, soil, and fish intestines. If environmental conditions are restrictive, Clostridium botulinum will produce endospores. These dormant structures are extremely heat resistant and will easily survive typical fish smoking processes. Luckily, botulism toxin is only produced when endospores germinate and the resulting cells multiply or grow. Clostridium botulinum will only grow under specific environmental conditions. These conditions are 1. The absence of oxygen. 2. A pH above 4.6. 3. A temperature warmer than 38 degrees Fahrenheit. 4. A water phase salt concentration below 10%. And 5. Relatively little competition for nutrients from other bacteria. Unfortunately, all of these conditions may be present in smoked fish that is not properly processed and stored. To make safe hot smoked fish, you must kill Clostridium botulinum cells during smoking. You must also prevent the germination of Clostridium botulinum endospores by brining to increase the water phase salt concentration. And you will prevent growth of cells after germination by storing the smoked fish at temperatures less than 38 degrees Fahrenheit. You may store your product under air to further reduce the risk of Clostridium botulinum growth. To make safe cold smoked fish, you will not kill Clostridium botulinum cells by heating. The cold smoking process is designed to avoid killing the bacteria that will outcompete Clostridium botulinum and prevent its growth. Let's look more closely at some of the control measures that we've just mentioned. If the water phase salt concentration in smoked fish was 10% or higher, no additional preservation steps would be necessary. However, most consumers would consider such a product too salty. So, a combination of refrigeration and water phase salt concentration must be used to ensure safety. Water phase salt concentration is controlled by brining or dry salting. The water phase salt concentration is measured in the loin muscle, which is the thickest part of the fish. Most fish processors hire a qualified laboratory to measure water phase salt concentration in finished product samples. The results are then used to validate the brining procedure used. The FDA recommends that smoked fish to be packed under air have a water phase salt concentration of at least 2.5%. Some states may even have more stringent standards. Michigan requires at least 3% water phase salt. If the smoked fish is stored under vacuum or a modified atmosphere, it must have at least 3.5% water phase salt. If the smoked fish contains at least 100 parts per million sodium nitrite, the water phase salt concentration must be at least 3%. Michigan also requires processors to obtain a variance for packaging smoked fish under vacuum or low oxygen atmosphere. A variance is also required if additives such as sodium nitrite or potassium chloride are combined with salt or used as a salt substitute. Be sure to check your state's regulations. Several factors during brining affect the final water phase salt concentration. So, there is no easy formula for setting your brine conditions. The key factors to consider include 1. Brine strength, 2. Brine temperature, 3. The proportions of fish and brine, 4. Brining time, 5. The size, form, and species of fish being brined, 6. The freshness of the fish, and 7. Whether the fish have been previously frozen. The salt concentration in your brine can be controlled by accurately measuring salt and water, as this plant does. Or you can measure the brine percentage using a salimeter, as shown here. If you use a salimeter, be sure to periodically check it for accuracy. You can do this using a standard salt solution. The brine should be kept cold to ensure that salt-tolerant spoilage microbes do not grow. State regulations may specify the brining temperature. For example, in Wisconsin, brine must be 38 degrees Fahrenheit or colder. In Michigan, brines as warm as 60 degrees Fahrenheit can be used for up to 4 hours. For longer brining, the 38-degree Fahrenheit limit applies. Records of brine temperature should be kept to document control of this step. In a given amount of brine, only a certain amount of salt can diffuse into the fish. If too many fish are put in the brine, the water phase salt concentration will not reach safe levels. If too few fish are put in the brine, the water phase salt concentration will be higher than is necessary and may affect flavor. Therefore, it is very important to standardize the weight of fish that is added to a given amount of brine. There are no regulatory limits on the duration of brining. Use test batches to determine the time needed for brining each size, form, and species of fish you intend to smoke. Many processors brine from late in one production day to early in the next day. In this case, brine strength and batch size are adjusted to ensure the correct water phase salt concentration is obtained. Fish being brined should be as uniform in size as possible. Carefully sort fish by size and brine each size group separately. Recently harvested fish do not absorb salt as quickly as those that have been refrigerated overnight and those that have been previously frozen and thawed. Your brining process should account for this variation too. For safety, every brined fish must have high enough water phase salt concentration. Don't take chances. Run some test batches for each type of fish and have the largest smoked fish in each batch tested by a laboratory. Write down the conditions used and keep the lab results on file. Be sure that you do not deviate from the conditions that gave acceptable results. Every batch should be monitored to be sure that the necessary conditions are met, and the results of monitoring should be recorded. This plant has a list of the brine percentages needed for a given weight of several types of fish. The volume of brine is standardized, and a specific weight of fish is added. Records are kept of the brine percentage and temperature at the beginning and end of brining. Periodically, have samples of each type of smoked fish tested to verify that your process is still achieving the safe water phase salt concentrations. The sample that is tested should always be the largest fish in the batch.